Our next speaker will be Yanting Shen, and she's at Kansas State University and is this year's winner of the Hans Konitz Award, the AOCS award that we give out every year, so congratulations. She will be presenting her poster, which is Modulating Intra and Intermolecular Interactions of Pea protein isolate to improve its functional properties. Yanting?
Yes. Hi, everyone. My name is Yanting Shen. Today, I'm going to share my research about the modulating inter and intermolecular interactions of pea protein for its functional, improve its functional properties. Protein denaturation occurs along with the reactions to cleave the covalent bond, peptide bond, or break down the hydrogen bond or hydrophobic interactions. The sodium sulfate is a reducing agent that can break down the protein disulfide cross-linking. It will result the free SH groups. The urea denatures protein through the dehydrating protein molecules, and it will cause the repulsion between the protein by hydrophobic interactions and competitive hydrogen bonding. But the urea breaks down the hydrogen bond more efficiently than affecting the hydrophobic interactions. The SDS dominantly break down the hydrophobic interactions, and protease break down the peptide bond and result in the smaller peptides. So for this project, the objective of this study was to investigate the functional properties of pea protein isolate by modulating protein covalent and non-covalent interactions and understand the physicochemical characteristics of the modified pea protein that are responsible for the functional change. So let's see our results. For the free amino group, we can confirm that the trypsin hydrolyzed pea protein had the highest free amino content. And for the free SH groups, with 1% of the sodium sulfate, the free SH content significantly increased compared to the control and other modifications. And for the surface hydrophobicity, 0.5% of the SDS modified pea protein exhibit the highest surface hydrophobicity among the others. The hydrophobic tail of SDS interact with the hydrophobic cones of the protein in the water, so it will result the largely exposure of hydrophobic side chains. For the SDS-PAGE under the non-reducing conditions, it is obviously to see that the protein modified by the sodium sulfate had overall lower molecular weight compared with others. And let's see the DSD result. The 1 mole of the urea modified pea protein had the highest denaturation temperature than the others. So that implies that the hydrogen bonding plays a dominant effect on the protein thermal stability. So next, let's look at our functional properties results. For the urea and SDS modified proteins have significantly higher values on the water holding capacities as well as on the emulsifying capacity and the stabilities. For the sodium sulfate modified protein have no significant improvement compared with pea proteins. The trypsin hydrolyzed protein have significantly lower values on the water holding capacities, but it has greatly improved the solubility at the pH at the isoelectric point, as well as good forming capacities. But for the oil holding capacity, all of the modifications have improved the oil holding capacities. And for the least gelation capacities, except for the sodium sulfate, the other modification methods are going to get a similar or even better gelation capacities than compared with control pea proteins. So last, we did the principal component analysis to further determine the relationship between functional physicochemical properties with different modifications. So we found that improving the water oil holding capacities, emulsifying properties, and solubility at pH 7 are associated with the non-covalent interactions. The forming capacities and the solubility at isoelectric point improvement are associated with the enzymatic hydrolysis, which is attributed to the smaller peptides formed through the hydrolysis and greatly unfolded the protein peptide chains. In addition, breakdown the covalent cross-linking is associated with poor gelation capacities that is due to these associated the globulin protein and inhibit the disulfide exchanges. So in conclusion, this study provided the fundamental knowledge of the protein functionalities related to the protein covalent and non-covalent interactions, and it will contribute to the broader field applications of P proteins. And that's all. Thank you for your listening. I'm happy to answer any questions.
Thank you, Yanting. A very good presentation. You've obviously done a lot of work. So
thank you.
I was wondering about your P protein isolate, and I will be revealing my ignorance here. Do you expect trypsin inhibitor activity to be present in P protein isolate?
Your question is trypsin will inhibit the activity. What kind of activity?
No, I'm sorry, let me be more clear. Many of these pulses and grains and oil seeds have a particular protein in them which inhibits trypsin. So that has to be measured with a trypsin inhibitor assay. And I wonder about P protein isolate. Do you know if that would be expected to have trypsin inhibitor activity in it, so that when you added the trypsin, it would have to titrate the inhibitor and then start working?
Oh, for this part, we haven't think about that, but maybe for our next step, we can do more research on this one for the nutritional aspect. But we did the digestion for these modified proteins.
Okay, thank you. Judges, do you have some questions for Yanting?
Yeah, probably I can be first this time. Thanks for the great presentation, and I like the idea behind this project because there's already a lot of study work on understanding the relation between secondary structure and functionality. I think your work provides new insights on this. My first question is, because the hydrogen bonding stabilize the secondary structure of protein, especially the alpha helix and the beta sheet, right? So my question is, why after using the urea to break down the hydrogen bond, but compared to the control protein, the secondary structure, including the alpha helix and the beta sheet, increased after this modification?
I think first maybe because of the concentration of the urea is not very high for this study, it's still low, and they have the, also competitive the hydrophobic interactions for the urea modification.
Okay, so my next question is, do you think this denaturation is reversible or not? Because after the modification, you use the dialysis to remove the chemicals. Do you think the interaction will reform?
Yeah, I think it may be reversible, but it will not be reversible after the heating process.
Okay, thank you.
Hi, Yanting. Thank you for this very nice presentation. I have two questions. The first one is about the protein hydrolysis you made. Do you know how far did you go for the hydrolysis in terms of percentage of hydrolysis? How many bonds did you break during this treatment?
How the bond breaks still?
Yeah, when you make the protease action, you cut bonds between the protein.
Peptide bonds.
Do you know how many percent of the total bonds did you break? Was it a very deep hydrolysis or very slight hydrolysis?
Oh, I think it's a deep hydrolysis. It can be proved by our analysis from the free amino group. So if you compare with the control, So it have the I remember it's a 54 the mmols per grams of protein will be released for the free amino group compared with the control is only 11.
Okay, thank you. And the second question is, of course, you made a lot of lab experiment, but do you already imagine how you can translate this into a food processing application?
Yes, actually, this is only the fundamental experiment, but we hope to further use it in the food application. For example, the disulfide, the cross-linking. So if we use sodium sulfate to break down the cross-linking, we can use other food degrade reducing agent to treat it in the food and make it more different structures. For example, in the bakery industries, use like the cysteine to treat for the cookies and make more spread ratios.
Thank you.
Thank you, Yanting, for a very interesting presentation. I think your research is pretty fundamental, as you mentioned. So I'm going to ask you a very fundamental or basic question. Can you tell me why urea can break up hydrogen bonding in protein?
Why the urea break down the hydrogen bonding?
In protein, yes. Because that's the approach you used to damage the hydrogen bonding in protein, right? So why you're using the urea can disrupt or damage the hydrogen bonding in the protein structure?
Because in the previous studies, in the chemical studies, they all use the urea to break down the hydrogen bonding.
Oh, okay. So you refer to the literature. Okay, thank you. Thank you.
So yes, it's very important to connect your results and your research now with what's gone before. So that's a very common. Are there any further questions? We have one question from the audience, which is similar to one that Denise asked, which was, do you have thoughts on the practical application with all the chemicals for food grade pea protein? Let me start over. Any thoughts on practical application with all the chemicals for food grade pea protein processing? I would reinterpret this question as saying, can you use SDS in food? Can you use urea in food?
No.
Okay, so these are fundamental studies about the effects. Yeah.
Yes.
But trypsin you could use, right?
Yeah, trypsin we can use.
Okay. Well, thank you very much, Yanting. I think
you.
we are finished with our questions.